This video, we're going to take a look at nucleic acid, the only molecules that can produce exact copies of themselves. There are three types of nucleic acids: DNA, deoxyribonucleic acid; RNA, ribonucleic acid; and ATP, adenosine triphosphate. Now, if we look at their basic structures, they all have similar components. They all have a phosphate group, they all have a pentose sugar, and a base attached. So let's look at the structure and function of each of them in a little bit more detail. So in terms of DNA, its job or role is to store and transmit genetic material in living organisms. It undergoes direct synthesis of new proteins and organic molecules. The main structural difference from the other nucleic acids is in the pentose sugar. Now, its name is deoxyribonucleic acid. Well, it's a ribose sugar, that's the pentose sugar, and deoxy implies that if we look here attached to carbon number 2, instead of a hydroxyl group, there's just a hydrogen, deoxy, lack of oxygen. Otherwise, it is structurally the same as RNA. So make sure to note the structural differences between the three. RNA is responsible for translating DNA into functional proteins. There are several different types, and they are denoted by a lowercase letter in front, such as tRNA or transfer RNA, messenger RNA, mRNA, ribosomal RNA, rRNA. The main structural difference here, again, comes from the pentose sugar. The name dictates ribonucleic acid, so it's a ribose sugar, and noticing it has the full hydroxyl group, whereas DNA, deoxy, is missing the oxygen on that carbon number 2. ATP, its job is to store potential energy within the bonds, within those phosphate bonds that can then be released and converted, the molecules converted to ADP, and that energy can be used for other biological processes. So when we look at here, there is the base, the sugar, but in this case, ATP triphosphate. There are 3 phosphates. However, when you take a phosphate off, energy will be released, and it will now be ADP, adenosine diphosphate, because there are 2 phosphates. There is also AMP, adenosine monophosphate, if you take another phosphate off. So in terms of nucleic acid classification, the monomers are nucleotides, but DNA and RNA are technically the only true polymers because they can become very long chain. ATP is not a true polymer because when you look at it, it can only have 3 repeating units, not technically a polymer. So let's take a look at the general structure of the nucleotide, or the monomer of a nucleic acid. 3 parts: a nitrogenous base, a pentose sugar, and a phosphate group. So let's take a quick second, make sure that we can draw this structure. When we're drawing a nucleotide, we start off drawing the pentose sugar. So we start with the oxygen at the top, 5-sided ring structure. It's through this structure that there is another carbon coming out, which attaches to the phosphate group. Remembering that a phosphate group attaches through an oxygen. So we draw our phosphate with an oxygen on either side. Phosphorus must also have a double-bound oxygen. Any of these oxygens out on the edge are negatively charged because they were once hydroxyls, but they have lost their hydrogens. Going back to the pentose sugar, we then need to add in the nitrogenous base, or a ring structure, and there you have it, a nucleotide. So as you saw in the drawing, we're going to have a nitrogenous base, a pentose sugar, and a phosphate group, no matter what type of nucleotide we're dealing with, one for DNA, RNA, or ATP. So let's look at the polymers then, DNA and RNA. So they're nucleotides, as we've mentioned several times. The difference comes from this pentose sugar. DNA having just a hydrogen attached to carbon number 2, RNA having a hydroxyl group. Each of these structures then has a nitrogenous base attached to it. Both DNA and RNA have these 3 nitrogenous bases: adenine, guanine, and cytosine. They differ in their 4th nitrogenous base. DNA has thymine, where RNA has uracil. We then take a look at these nitrogenous bases, and we want to look at whether they're single or double rings. So adenine and guanine are called purines. They have double rings, or 2 rings, as part of the nitrogenous base. Cytosine, thymine, and uracil, on the other hand, are called pyrimidines. They have a single ring each. So how do we go from these nucleotides to actually making the polymer? Well, we take a look at the nucleotides, and it's the phosphate group of one that interacts with the hydroxyl group of the next. And just as we've seen in other dehydration synthesis, we have the loss of a hydrogen and a hydroxyl, which will result in a covalent bond. So the phosphate of one nucleotide links to the sugar of the next via a condensation reaction, meaning that we have the production of water. So as this hydrogen and hydroxyl get cleaved off, we have an oxygen and a carbon that need a bond. They bond together, and we call this a phosphodiester bond. So when we're making a polymer of a nucleic acid, it's held together by phosphodiester bonds. Take a moment now to make sure you can draw two nucleotides attaching together through a condensation reaction. So then how do we actually get to DNA? So these phosphodiester bonds form a sugar phosphate backbone. The nitrogenous bases then stick out the side. They are held together via hydrogen bonds. Now there's a specific way that they have to go together. In terms of DNA, guanine will always partner up with cytosine, and these are through hydrogen bonds. Guanine and cytosine will always form 3 hydrogen bonds between them. Adenine and thymine, on the other hand, always bond together through 2 hydrogen bonds. One way you can remember it is the A and the T, the straight line letters, stick together. G and C, both curved letters, they stick together. This is called base pairing, and you will always find G bound to C, A bound to T when you're looking at DNA. So that would be how those 2 strands of DNA are held together. So DNA actually has 2 strands. There are 2 sugar phosphate backbones, each of which having nitrogenous bases coming out in the middle, which are hydrogen bound together. So hydrogen bonding holds the strands together. Covalent bonds holds that backbone together. When we're talking about RNA, though, it's a single strand only. So DNA has the hydrogen bonding holding 2 strands together. RNA only ever has 1 single strand. Now, the other thing I want you to notice looking at this diagram is DNA is said to run anti-parallel. Well, if you notice the phosphates and the pentose sugars on this left-hand side, they're right side up. This other side is upside down. So anti-parallel means that one side is flipped upside down from the other. Now, we've all seen that characteristic double helix shape of DNA. The reason it twists is because these molecules are actually 3-dimensional, and in order for them to fit together like puzzle pieces, there has to be a twisting as they go down due to the structure of the nucleotides. So that is why DNA is called a double helix. It's because there are 2 strands and it twists because of the shape of the nucleotides.